Welcome back friends. Welcome to another video tutorial from Shomu's Biology. And in this video lecture, we want to talk about patch clamp technique. I actually got this request from many of my subscribers to make a video on patch clamp technique. So this is for you. What is patch clamp technique? And mostly we are going to see the overview about this technique. And we'll see the importance of patch clamp technique and why we do that. Patch clamp technique is an electrobiological technique, or it's mostly like a idea of measuring electron potential or membrane potential across the neurons. So mostly this technique is used for the neurobiology or neural science. In that case, to understand about the neural transmissions between those cells, as well as about how a neuron is reacted by the release of specific neurotransmitter and stuff. That's the idea about this patch clamp technique. Now what we do exactly, to understand patch clamp technique, you should know and you should have a basic idea about how those nerve cells work. Now for every single cell in our body, we know those cells have cell membrane and they have different ion channels embedded in the cell membrane. So let me start with writing some easy fact regarding the normal cells and also the neurons. So if you look at the cells of normal cells and neurons, all of them have those ion channels embedded in the membrane. Different types of ion channels, mostly some of them are potassium channels, some of them are sodium channels. Calcium channels are there, chloride channels are there. Different channels are embedded in the membrane. Now for a neuron, the number of these ion channels are more compared to the other type of body cell. Now as they have multiple number of ion channels, the importance of these ion channels is to periodically open and close so that a specific type of ion can go in or out of the cell. Now why a cell need that? Why a cell need for them ions to pass, positively charged or negatively charged ion to pass through across the membrane? What is the importance? The thing is, cell membrane, and actually all the cells we know in our body, they maintains a membrane potential. We call it as a resting membrane potential. Okay, this resting membrane potential for normal type of cell is minus 90 millivolt, while for neurons is somewhat like minus 70 millivolt. That is the range of this resting membrane potential. That means this membrane has a voltage across its membrane. This voltage is sometimes minus 90 or minus 70, and that is the base level. So we call it when the cell is in the resting state. Resting state means the number of ions passing in and out are mostly balanced and they have this voltage completely constant. But for the neuron cells, what happens? This voltage gets altered. Why a voltage will be altered? If a flow of current is there, voltage will be changed, right? That's why the voltage is altered. Whatever idea we have about the electrical wiring and stuff, we know that current flow can change the voltage. So here in this case, flowing of this sodium or potassium or chloride ions across the membrane can alter this resting membrane potential. So that is the importance for the neurons. So let's say ion channel opens or closes. All these things lead to change in the resting membrane potential. And this change in resting membrane potential, the change in the voltage across the membrane of neurons, can be measured. Now these changes are important for transmitting signals from one neuron to the next neuron, because neurons are connected. They form a long distance route. Even there are large neurons starting from our central nervous system and it's going to the tip of our toe. So it's large, long neurons. So those neurons are normally interacting like one neuron attached to axon of one neuron attached to dendrons of the next neuron. By this way, changing of this chemical signaling due to any response to any kind of neurotransmitter like acetylcholine, they start changing all this ion flow in and out, and that ultimately results in the modification of the membrane potential. So once this membrane potential is modified, that suggests us that that neuron is transmitting some signal, right? So one neuron is transmitting a signal. So whenever we call about the nerve impulse, it's nothing but change in the resting membrane potential, and we call them action potential. Now in this video I'm not going to talk about action potential in detail, because if you want to know about action potential or resting membrane potential or in details about the ion channels, I'll recommend you to watch my new videos about the human physiology, in the nerve physiology, when I talked about all of these things in details. So as you know this part, because this is required to know the whole process and technique. Now the question is, how come we know that this ion channels opens or closes? We know that when sodium channels open, we know when potassium channel opens, when sodium channel closed, because those channels, those ion channels are sometimes controllable, like they are called as voltage gated ion channels. So due to the change in the voltage, the channel can open or close. So how do we know about all this stuff? And let's say we are doing the same research not only for the neurons, but we can also do this research for the cardiomyocytes, okay, all these tissues, or any normal type of muscle cells. In those cases, what we can say, that the movement of all those ions are controlled by the voltage change, and this change of voltage required for the movement of the sodium, potassium or chloride to flow in or out demands specific modifications. And how can we know what kind of modifications are required for the signaling to pass and work? To understand that, we utilize a technique called patch clamp technique, which will give us an idea about the activity of a specific ion channel on the surface of a membrane. Remember that. So it gives us an idea about the activity of either one specific ion channel, or we can also utilize patch clamp to get an idea about the whole cell, and how a whole cell will behave in response to this opening and closing of ion channels. In response, sometimes considering the voltage to be constant and getting the idea about the current, sometimes current to be constant and getting the idea about the voltage. So all these things we will understand, and both these things are required for understanding the patch clamp, for understanding this idea with the patch clamp technique. So we can find that out easily with patch clamp technique. Now what this technique is suggesting? Now in your mind you can think, what can be the technique that can give us an idea about what's going on across this ion channel? So definitely we need to extract the ion channels. Now the problem with this idea is, if we extract a part of this ion channel, so extract a fragment of cell membranes, that can be dangerous because the cell will ultimately die. If the cell dead, in that case there is no way we can measure the activity in real time, because we want to know the sequential events, because you know the process of action potential, generation of action potential, propagation of action potential, it demands sequential stages from the start to the end. Now the sequential stages will be tightly regulated by different opening and closing of different ion channels. For that to know, we need to do it in real time. You cannot simply scrap open some part of the ion channel and check it. It's not possible. So what is the technique that can be used to measure the activity across a single ion channel and in real time? For that, what we do, we take a tissue slice, or usually we use cell culture, the animal cell culture. For example, we are looking the same situation in the mouse neuron. So we take the culture of this neuron cells in the plate, we put it under a microscope. Those cells are tiny, we cannot see them separately. So we put it under the microscope. And you know, once we look cells in the microscope, you will see all the cells clustered together. But we want to study a separated cell. We need a complete unit cell, not attached with any other cells, to measure this whole process. So this is another important thing, that we need to find the isolated cell in that plate. Once we find this isolated cell, let's assume we found out an isolated, let's say this is a nerve cell, we found this isolated cell. and this isolated cell there are different ion channels embedded. Okay, and let's say another ion channel here, and let's draw 3 different ion channels with 3 different colors. It's embedded. Now what we do, we find out a specific ion channel to tag with, but actually it's very difficult to find out a specific ion channel. But what we do, we simply use a pipette, because the whole idea about the patch clamp technique to use a pipette, which is very thin diameter, it's very thin diameter of pipette that is kind of inserted in those area of the cell, so that the pipette is in direct contact with the surface of the cell membrane which carries the ion channel. Let's assume here this is the pipette, and the mouth of the pipette, so this is the pipette, let's say the mouth of the pipette is smoothed down with heat. So we heat this pipette so that the end or the mouth becomes very smooth, so that it can properly interact with the surface of the cell membrane. Okay, so that pipette will be in direct contact. And then what we do, normally this pipette, inside the pipette is filled with solutions which are containing conducting particles. Okay, solution with conducting particles, and there is a suction attached at the end of the pipette. So like any other pipette, there is a suction force out there, but the suction force is controllable, we can control that. And there is also an electrode inserted there. Actually the electrode is embedded to an amplifier. Okay, and also that amplifier is another domain that's going to the ground. And then finally, throughout this amplifier, ultimately we have the signal recipient, that will the receptor that will check the response in the screen. That is kind of the circuit for this, the simplest form of the circuit that we know. One electrode is inside this pipette, one electrode is in the ground, and it's attached to an amplifier, and there is a resistor out there in the middle right after the amplifier. Amplifier is also connected to a resistor here. Okay, now the idea here is very simple. We put this pipette, we know the pipette is going to properly interact to the membrane. We create a little suction there. Once we do the little suction, the pipette is properly attached airtight way to the membrane. So as it is attached properly to the airtight way to the membrane, that part of the membrane, if it carries any ion channels, that will also be internalized there. It's properly tagged and blocked there. So the portion of that ion channel, the extracellular region for that ion channel, becomes this solution that is inside the pipette. Rest of the thing is inside the cell. So now what we are looking, the extracellular part for this. So forget about rest of the part of the cell, because we want to find out the effect of a single ion channel. So now this is the scenario where we have the ion channels attached to the pipette. Inside of the ion channel is the cell cytosol. Outside region is the conducting part. Now what we see, if this ion channel opens or closed, in that case opening and closing of this ion channel transfers, let's say it's a sodium channel, so sodium movement will be there. So movement of the sodium across this membrane can change the voltage, and we can measure the voltage here at the end, right? Now the question is, once we add this pipette, normally the cell is having a specific membrane potential based on outside and inside. Think about it. Whenever we attach this pipette, I'm telling you that this pipette, the internal region of the pipette carrying the conducting particles, becomes the extracellular part for that specific ion channel of our target. So what it does, the earlier membrane potential which was maintained by this ion channel gets altered whenever we attach the pipette. It's altered. So to make it balanced, what we need to do, we need to also add some other energy or positive or negatively charged particles here in the uh, balance that current, so that the voltage becomes steady again. So whenever, if you think, whenever you add this pipette at the beginning of this experiment, you will see, if I look at the data in the voltage, how it will look like. It will look something like that. The voltage is in resting potential, then slowly comes down, and then again it will go back to the normal state. So that's what happens all the time. So this falling of the voltage, if you look at here, that is due to the time when the pipette is being attached to this membrane, and specifically to ion. Whenever the attachment is proper and there is a very airtight attachment, due to the suction force and suction pressure applied there, it will be properly attached to the membrane and be very tight over there. So the resistance over here will be very less. In this particular case, the resistance will be in high, not actually less. Sorry, it's in giga ohm. So the resistance will go to the giga ohm. As a result, for that, you will see that thing will come back to the normal membrane potential again, the ground level. Now the question is, one electrode is embedded in the pipette, the other one is embedded to the ground. Why so? Once the other electrode is embedded to the ground, that two electrode is going to give us the idea about the zero level for the current. Okay, because if we simply measure the current for this inside the pipette, that will be varied. So we need to have a base standard to measure the change in the current due to the intracellular events. That's what our idea is. We know what current is there in the pipette, but it's modified. So the best way to cancel this out, and the only way to make it a balanced electric current is simply put another electrode to the ground. So now we have a base or zero or ground state for the current, and that is zero. We call it zero. That's why we have a ground. And the phenomena, while this pipette is properly attaching due to the increased giga ohm resistance over there, and we call it as a giga seal, which is allowing this pipette to properly attached with the cell. Now once it's attached to the cell, everything is properly finished, it's there. Now if there is any, by any means, inside the cell, with time we will see this experiment, if there is any way this channel opens, sodium start flowing out, for example, sodium start flowing out side of the cell into the pipette. Let's say this red dots are sodium, it's flowing out for a while, and then again this channel closed down. So in that case, what will happen? Membrane potential will alter. So the signal that we see here will also alter, because earlier whatever thing was mediated was balanced to zero, but now due to the movement of the sodium, what we know, that the outside will be slightly more positive, inside will be slightly more negative, so it becomes negative. What we can see, for a specific time duration, we can see the opening or closing of a target membrane ion channel to say whether molecules are going out or going in with that time, and how the voltage is going to be altered for that. Now this process can be repeated for two different approach. One we can do is we can balance the voltage. We can keep the voltage constant by constantly supplying other electrolytes inside, positive or negatively charged ions inside of the pipette, but we will measure the change in the current across the membrane. Or we can do another thing, we can alter, we can make the currents constant. We can apply some current and make it constant from both across the membrane and see the change in membrane potential. That will prove us the effect of any third molecule that influence membrane potential or the transfer of ions across the membrane. Now normally the current alteration provided by one single ion channels are very low. So that is the reason we should have a very ultra sensitive amplifier and detector to change to find out the change in the current. It actually goes like in 10 to the or so, this modification. So it's even picoampere level. It's very hard to find out with normal type of amplifier that. So all the setup and machineries that we need are very much sophisticated, And this is not only a job of for a biologist. That person who will handle this must know all those instrumentation and structures, and how electric current works, how voltage works, how to measure them, and how to find out the graph. And also this is delicate because this pipette is interacting with a cell, and you know how tiny it is. That diameter is very very less, like 5 micrometer or 1.5 to 5 micrometer range in the diameter for the pipette, and that will be very difficult without a properly trained eye. Normally we have a trained system. It's a dispenser or pipette dispenser over there that will help the pipette to move accordingly. It's not a human hand, because a small fraction of vibration can destroy this whole experiment. And another thing, while the experiment is being performed, we use non-vibrating table and non-vibrating table microscope, so that everything is without any single vibration. If there is a little vibration, the whole experiment will be ruined. And the electrodes that we use normally for this process are also very expensive, so this whole experiment is also a little expensive. Now in this case, the electrodes used are silver electrodes, silver chloride electrodes that is there. And so it's a very expensive one experiment. This requires very much proper professional skills to handle this experiment, and also it requires, but the data analysis is not that difficult for them. It's quite easy compared to the other data analysis part, but you also need good softwares to manage and tag it with all these facilities. So these facilities are mostly available in the pharmacological conventions in pharma industries. While we want to know about different chemical factors that can block different ion channels or help opening different ion channels in different diseases and situations, because many of our diseases rely on the problem with ion channels opening and closing. So if we find out which molecules inhibit this ion channel or opens the ion channel in response to time, we can develop specific drugs. We can target ion channels with different drugs, and actually you will find huge number of ion channels inhibitors as our drugs, as medicines that we use these days. And very easy type or simpler type of one, their photon reflux inhibitors are very much common drugs that we use, all the type of drugs that we use very commonly in our life. So similarly, for example, a type of process or a problem or disease associated with rapid influx, a rapid efflux or something of some ions and some chemical factors. So if you block that efflux pump, that can prevent us from the disease. So what we do, we find that out using patch clamp technique. Without patch clamp technique, we'll never know which ion channel is opened, whether the efflux pump is working or influx pump is working or not. So these are all the ideas and advantages of using patch clamp technique. But this is not the end yet, because this type of technique that I mentioned, this is only one type of patch clamp technique, which was earlier discovered in 1970s. So it's not a very new technique, but still it's being developed rapidly since then, and now it takes new heights. Why? Because earlier the technique I mentioned, it's simply a whole cell while a pipette is interacting with one channel. This is known as on cell patch clamp technique. On cell, because the patch clamp, because in this case simply the pipette is contacting the cell directly on top of the cell. Other variations of these techniques are also there to study a specific ion channel or to study whole cell. Remember, I also told you we can study the modifications of the ion channels throughout the whole cell. That will give us an idea about what are the modifications and how the cell is working. For that process, we have the modifications, similar things, and this pipette is in contact with the cell. But in this case, what will happen, the force, the suction pressure that we apply, that is huge, and due to the huge suction force, the part associated to the membrane gets cleaved. So what happens actually, if I draw it sequentially, let me draw it from here, the different modifications. So in this case, what we have the cell. Let's say this is the cell, and let's say this is the pipette we have. Now it's interacting to the cell. This is on cell type. Now the second one that I am going to talk, this is for whole cell. While this pipette is in contact, the pipe or the pipette allows some part of the cell to rupture over there. So the content of the cell is now completely released into the pipette itself. So the cytosolic component, if you see, these are the cytosolic component, that thing is also inside the pipette. So what's going on here is, inside the pipette, it's very same like the cytosol. So the cytosol and the pipette will contain similar materials inside. This is known as a whole cell patch clamp technique. And this technique is going to give you an idea about how the cell behaves. All the cell is behaving due to opening and closing of all the ion channels that are present on their surface. Because now you're increasing the area of the intracellular space, or intracellular regions, or cytosolic region of the cell. Now, another type of modification is there to study a single ion channel is simply, again, we apply a little force, and applying little force will cause some part of the membrane to rupture, and the part which ruptured remain attached to this pipette. So force applied, ruptured, so membrane gets ruptured, but the part of this membrane, if I draw it here, let's draw it a little big so that you can see this, and the part of this membrane remains attached to the pipette, and let's say an ion channel is placed in the middle. Are you getting my point? There was an ion channel over there which is completely ruptured, and earlier in case of whole cell goes away, but in this case it's something like ion channel, part of the cell membrane, and then rest of the mouth of the pipette. This is the structure. So ion channels inside, so because earlier, once the ion channel is embedded in the membrane, if I draw the normal structure of the ion channel here, try to find out the difference. Normally ion channel is embedded in the membrane. So you see, it's completely cleaved out. So whatever thing was inside, now it gets outside, because it's now outside in the air. So the pipette is in the air, so inside of the ion channel is now contacting air. So actually it's not only contacting air, but also we can put it in different medium containing different number of positive and negatively charged ions, to find out the function of this ion channel only. And also we can find out removal of this ion channel, what impact this rest of the cell can achieve. Both of these things can be understood with this process. So this is a very important way to find out the functionality of a single ion channel. This is known as inside out. Inside out. And the last one, another variant, it's also similar. This is a cell, and again the pipette. Again, what we are doing here, we are simply doing for the whole cell type of situation. And while we are doing whole cell, some part is there which is cleaved. Let's say here it's cleaved. Some fragment of the cell membrane is cleaved like this. So it's completely cleaved. Normally trying to create the whole cell extract, but we applied a little more pressure there, that will also rupture the membrane from both the sides. Let's say do the same thing, but if it ruptured both the sides, it will give us this situation. So in this situation, what we have a ion channel attached in one part, and then what it does, actually the two terminal of this membrane can reorient, because they are phospholipids molecules, they can re-enter, reorient. By some means, if they reoriented, what it will form? It will form a structure like this, a loop like structure. Okay, so here we see it form a loop like structure. Now in this loop like structure, if you remember, this was the outside. So the outside of the ion channel is present outside. Earlier in this case we saw that outside was in and the inside was out, so we call inside out. But in this case, outside of the ion channel is in outside port, so we call it outside out. Now why we need this kind of situations? So the difference between inside out and outside out, if you are confused about this was the inside out, but the structure of outside out will look something like this. And this is the ion channel. This is outside out, this is inside out. The modifications for that, both of them are required to study single ion channel function. But inside out is required to find out most of the ion channels how they behave, while for outside out type of patch clamp technique is required to study the movement and modification of a specific ion channel in response to some chemical factors. So if we think like this type of ion channels, if we suspect that this ion channel, let's say ion channel X, this ion channel is much important, or it has a role somewhat, it involves with the role of neurotransmitters, and we know how neurotransmitters work. It moves there, binds with some receptor on the membrane that allows the one specific ion channels to open. So if we suspect those kind of activities and trying to find out the effect of neurotransmitters in different types of ion channels, then outside out is the perfect way to go, because then we separate this fragment. Pipette becomes intracellular, this becomes extracellular, and then we allow all those neurotransmitters to act, right? If let's say this is excitatory neurotransmitter, allow the ion channel to open up or something like that. So if we add acetylcholine, it let's say opens the ion channel. So we can find out, yes, this is an excitatory neurotransmitter release, and this ion channel usually opens in response to the excitatory neurotransmitter. So these are the ways the variety develops based on the different types of patch clamp. So for understanding whole cell, we use this whole cell patch clamp technique. This is on cell, whole cell, inside out and outside out. Both inside out and outside out are required for understanding single channels. Whole cell patch clamp is required to find out, or open cell is also another process to find out the whole cell function of ion channels, while on cell ion channels or clamp techniques can be utilized to find out a single or a combination of ion channels activity. So we not only can find out the effect of neurotransmitters, the effect of different chemical factors and drug targets, but also we can also tag with this patch clamp technique with calcium mediated fluorescent microscopy. In those fluorescent microscopy, we allow the movement and flow of calcium and stuff, and the modification with that and change in the fluorescence color inside the cell or outside the cell. That will be also very beneficial to find out the whole process in real time. And actually you can see it as a screen, because everything we can how a cell is behaving in response to different chemical mediators, and that's a huge achievement. That's why this technique is very important technique, though delicate, and also requires a huge amount of professional skills to handle it, and also expensive, but still a very important technique in the field of neuroscience and neural biology. And the more we go on, this technique is more will be needed and developed. So I hope I am able to explain patch clamp in overall basic way. So if you like this video, please hit the like button, subscribe to my channel to get more videos like that, and share this video with your friends. Thank you.